Thank you all for inviting me. It's great to be here. So what I want to do today is talk about a whole range of issues that surround organisms, model organisms, and so on. So we're going to start off by talking about what model organisms are, why they're successful or not. Does model organism research actually exclude or the organisms? So does it drop research biodiversity? So in other words, has it forced out other kinds of research? Whether you should allow one organism to stand in for many. So in other words, do you actually need that biodiversity? And then this bigger question of what actually guides organism choice. And by the time we get to this one, we'll actually do some history and get into a really interesting example. But I want to start with this list. This is the National Institute of Health list of model organisms. It's what we call the canonical model organisms. Before 1990, when this list was made, the word model organism was used very, very infrequently. After this list comes out, it becomes sort of prevalent. Now it's used just promiscuously. Biologists call everything a model organism. I do not. I refer to these as the model organisms because they have a different status. They were named as part of the Human Genome Initiative in 1990. They were named to sort of buy the love of biologists who knew that a lot of their funding was now going to go to the Human Genome Project. So all of these organisms got priority to have their genomes sequenced. In fact, most of their genomes were sequenced before the human genome was sequenced. Okay? They're models in the sense that they're models for human biomedicine. Right? So not models in any other general sense. They had to have some direct payoff for the National Institute of Health. And we'll come back to that. It also explains why there's only one plant, Arabidopsis, on the list, because plants generally aren't like humans and generally are not good models for our health, right? So that's the sort of idea. Now, one of the major consequences of this was that hundreds of millions of dollars gets preferentially invested in this set of organisms, right? And this creates ill will among people who are not sort of in this list, right? And so what we decided to do is actually try to understand this question of, does being on this list actually give you some sort of preferential success? So in other words, did you really get some benefit by being named by the NIH as one of these canonical model organisms? So the way we interpreted success, just to get a very quick measure, was what's the publication rate? How many articles are getting published for each of these kinds of articles? And so using big data sets like Web of Knowledge, we were able to go through and actually create sort of some basic data on publication trends. It's a little dark, but I'll explain. So this is just part of it. The bright blue line is the oldest model organism we have. So it goes back there to 1900. It's Drosophila. And as you would expect, it does very, very well. What's paradoxical about Drosophila is you would have thought that we'd have learned everything about fruit flies a long time ago, but it's continuing to do extraordinarily well. Arabidopsis is this dark blue line. You notice it sort of takes off right here about 1990 and goes almost straight up. It now exceeds Drosophila genetics. When we published this result, the Arabidopsis people went crazy on Twitter. Like, we're better than Drosophila was all they had to say about it. The same thing with zebrafish, but notice this yellow line. This is Xenopus. So this is the South African frog that's used in some embryology. It's going down, and it's going consistently down, right? So being designated a model organism isn't a guarantee of success. In fact, Xenopus has not been able to get it together to become a successful organism. On this slide, I also include this red line, which is maize, corn. It is not listed as a model organism, but it's doing incredibly well. And so the question is, obviously, just being named isn't enough, right? So what makes these things successful when they're successful? If you ask a biologist, I was in a biology department when I did this research, they would say money. Money is the top thing that makes an organism successful. And if you look back at this graph, there are some deep correlates. So the two lines that are going almost vertical got hundreds of thousands of millions of dollars invested in them very quickly. So Xenopus and Arabidopsis, you can definitely see a very strong correlation. But that's not the case with the other organisms. So there's definitely some component to it, but it's not an automatic feature. Instead, when we started looking at the features that made all the systems successful, the ones that were successful, we found that they had community resources. And by those we meant they had developed certain kinds of infrastructures. They had online databases, they had stock centers, they had standardized protocols that made it very easy to talk to each other and communicate their results. They had training workshops, seminars, established protocols, very well-established means of communications. These usually started out as like newsletters. So Drosophila Information Service was the first one in the 30s. And they actually would have conferences just dedicated often to their organisms. So you go to the fly meeting, for instance, that kind of thing. The groups that didn't succeed, Xenopus, had none of these. Okay? And so the Xenopus white paper comes out, they set up their protocol at the Marine Biological Laboratory, but it kind of looks like it's too late. And it's too late in part because zebrafish, I think, has taken part of their research over. And they may not be able to build the infrastructure fast enough to catch up with the zebrafish community now. This is also why maize is doing really well. Maize also has all of these infrastructure features, and of course, you can eat it, which is good. People like eating it. You can eat fruit flies too, but it's not the same appeal, right? So we found that these features are some of the features that make it successful. These are parts of what Rachel Ankeny and Sabina Leonelli now call a repertoire. Right? So these are features that make certain organismic systems or certain experimental systems incredibly successul. They go beyond sort of this Kuhnian paradigm, which is just a purely intellectual construct, to actually talk about the kinds of things that allow research programs to flourish. Now, is it too much of a good thing? Did model organisms actually drive out other organisms? In order to answer that question, we went to the Journal of Genetics, and we read them all, and counted the organisms that were used in 5-year periods. And then we just divided them into the numbers of model organisms and the number of non-model systems. And so this is the sort of basic graph. You can see by basically 1985, about 75% of the articles in the journal Genetics are concerned model organisms. About 25% are non-model systems. This does not go to zero, right? So about a quarter of the articles are still about non-model systems. The majority, though, are with those new models. If you then apply some basic ideas from ecology about richness and diversity, but instead of applying them to species, apply them to species in the journal Genetics, right? So we call this research biodiversity. You get a little bit more refined. So species richness is just the number of species. And so you can see down here, the number of model organism species is never going to go above 15 because there aren't more than 15. But the number of non-model systems is doing quite well. This diversity index is just the Simpson-Gini index, so it just measures evenness of the distribution across the species category. And you can see that the non-model systems basically have a higher diversity. There's a wider spread. So in other words, it's not just a single article on petunias or a single article on turnips. It's actually a fairly solid set of non-model systems. So sort of contrary to the complaining that sometimes will occur among biologists, non-model systems have not been driven out of genetics. And in fact, the ones that are remaining seem to be very consistent, more heavily researched than you would expect. They're not rare variants in this literature. Okay? We're doing this for other disciplines now. We're doing it for developmental biology, which seems to have this exact same pattern. We're trying it also for cell biology. But cell biology, we discovered, doesn't actually list any organisms in their articles. They use a code, and it just makes it much more difficult. So which letters are capitalized in the gene name tells you which organism it's from. And you have to sort of know the secret code to be able to suss that out. Now, one of the major complaints about organisms is this presumption of generality. This idea sort of expressed by Jacques Monod, that's what's true for E. coli is true for the elephant. Notice E. coli is not on the model organism list. It is not a canonical model organism for anything, even though it's one of these established systems. This seems to irritate certain kinds of biologists. The idea that you don't have to actually study a diversity of organisms. You can pick a single species and sort of generalize very quickly from that. This sort of raises this question of, does research biodiversity actually matter? People have been pushing back at this, saying, look, E. coli doesn't even represent E. coli, right? because often with these model systems, they've been so standardized, so structured, that they are nothing like a wild variant anymore, even though you might call them a wild type, right? They are not wild in any sense. So standardization has erased a lot of that variability. But this is an even stronger claim, right? This is not a claim that you're just using a standardized variety. This is saying you don't even need to consider anything else except E. coli, right? So it's a much broader generalization across species that many people are objecting to. And in particular, people that are studying evolution find this to be an incredibly unrepresentative sample. So here's a pretty bad phylogeny. It actually gets worse if you put in the archaea, because it's a huge amount of biodiversity that's not considered. And you can see that the model systems, aren't covering huge parts of this tree. No plants, of course, except for Arabidopsis. So they are in no way phylogenetically representative. They don't represent ecological diversity, developmental diversity. And so if that's your guiding question, the model organisms are a poor choice for research. But remember, the NIH is about human biomedicine. Their guiding question is not ecology or evolution, right? So they were never intended to cover any kind of phylogenetic diversity. Nevertheless, once they're established and have this sort of position of authority, this problem arises, right? Now, this leads to this question of how do scientists actually choose their organism, right? Should you jump on the bandwagon of one of these model systems? It has all of the infrastructure ready for success. There's a community there, they'll train you, it's got resources, people that love to talk flies. Why not join the happy community of Drosophila researchers, as opposed to picking something that no one has ever researched before, okay? To sort of answer this question, we went back to one of the most famous sort of pieces of advice in the history of biology about organism choice. It comes from this guy, the Danish Nobel laureate August Croc. He wins the Nobel Prize for his work in physiology. And he has this statement: for a large number of problems, there will be some animal of choice or a few such animals on which it can be most conveniently studied. So this is called the Croc Principle. It was so named by Hans Krebs in the 1970s. And this is a meme on the internet, right? It is everywhere. Especially people who are trying to justify their organism choice seem to come back to this. And it has a whole bunch of interesting variants. And you can see like this paper from '92, they say for every biological question, there's an organism best suited to its solution, which is not exactly what this says, right? This is a much stronger claim. Every problem you have, there's going to be a single organism that's the best organism to study that. This leaves open the grounds for comparative study. This says no, right? And if the best organism is Drosophila, so much the better, right? So how do we figure out and sort of respond to this question? Well, one of the things that we've been trying to do is actually take it apart and trying to figure out what are the actual criteria that biologists have used in the past or might be using today. And so what we've been trying to do is unpack this idea of convenience and realize that that is not a super helpful description of the kinds of things that ought to come into an organism choice situation. So sort of a priori or based on our experience, we started to identify things that we think we've seen acting. So they include things like phenomenal access. In other words, in the system that you have, can you actually see or get access to the particular kind of thing that you want to study? And we'll talk about examples of that. Material availability is a huge one, right? Can you actually get to the stuff that you want? Is the cost reasonable or not? Ethical constraints play a huge factor here. So does tractability, especially when you take something into the lab. Is it viable? Is it durable? Can you standardize it? Will it respond in situ? So for instance, in marine systems, a lot of the animals that used are from estuaries because they're from brackish environments. They're very tough. They're hard to kill, right? And that makes a good lab organism. When your graduate student clean the tank like they're supposed to for 3 weeks, the fish are still there, okay? Training, tacit knowledge, how much of that is required. Institutional support can be huge. Are there methods available for analyzing it? What kind of community do you have to support you? Informational, so these are those databases, newsletters. And then this last one, opportunities for discovery, generalization, and comparison. In other words, does it have the sort of opportunity, the promise of new problems that you're going to be actually be able to do something with in an interesting way? Now, to sort of make this into a much less abstract kind of thing, I want to walk you through a case. And the case, we can talk more about how I picked it, because there's actually a big methodological procedure. But we used the General Embryological Information Service, which was like it was a phone book for developmental biologists from the 1950s through the 1980s. We've digitized that entire phone book and created a database of every developmental biologist in the world. And so this is how we find interesting people to work on now. And so we wanted to study a system that was a marine system. And so what we did is we looked for the most commonly used marine organism. It turns out it's echinoderms during this time period. And so then we looked at particular problems that echinoderms were most used for, and we landed on fertilization and cell cleavage that immediately follows fertilization. And then going down the list, we picked out two sets of names, the Colwins and the Dans, both couples, both who had done work at the Marine Biological Lab at Woods Hole, and then the Dans had also done research at Misaki. And so we decided to focus on this pair of couples to try to understand both how they came to do research on fertilization and cleavage, and how they picked the organisms that they actually picked. So a little background on fertilization, especially fertilization at MBL. The major player here is Frank Lilly. He's one of the founders of the Marine Biological Lab. If you go there, it's Lilly Hall, it's the hall you're going to go into. And he had a theory that the sort of reaction between sperm and egg is mediated by an external chemical that he called fertilizing. And the idea behind this, the evidence for this, is that if you put egg and sperm in seawater, they would sort of precipitate, they agglutinate, they coagulate together, and you can actually see it happening. And so we thought, this must be a chemical reaction, something like an immunological reaction maybe, and it had to be sort of facilitated by some external chemical. So he spends most of his life trying to find that chemical, and of course, never does, okay? But it sort of sets the stage for how people are going to do fertilization research, or a major part of it, in the early 20th century. He's using this marine worm, Nereus, for his work. His student, Ernest Everett Just, is doing the sort of the immediate next stage of biology, which is the cleavage once you have a fertilized egg. And he's also studying it in Nereus. Cleavage research up to this point is what I call primarily structural. They're looking for where does the sperm enter, what is the sort of geometry of cell cleavage, is it spherical or a spiral pattern, is it even, is it uneven? And so it's primarily descriptive comparative kind of work. So if you were going to go into this field, you would be reading papers by Just and other people who are describing that sort of at a very phenomenological level. These two people are important because all of the next four we're going to study all went to Penn, so University of Pennsylvania and Philadelphia, that all spent their summer at the Marine Biological Laboratory at Woods Hole, which is kind of like where everybody in the United States who is a biologist spent their summer. This is a nice place to go for the summer. So let me introduce you. This is Katsuma Dan. He called himself Katie Dan. And Gene Clark, who will become Gene Clark Dan. Katsuma Dan is from a very well-regarded family in Japan. It's a noble family. His father was the Baron Dan. His father was also head of the Mitsui Group, which is one of the largest conglomerations of industrial companies in Japan at the time. They run mining, railroads, steel. They're all kind of like Rockefeller, Carnegie's in the United States. His son, Katsuma, is very interested in marine biology, comes to the United States to study with Victor Heilbronn at Penn, earns his PhD in 1934. He's forced to go home before he actually earns his PhD because his father is assassinated, because his father is very pro-American. His father had earned a mining degree at MIT and is assassinated by a pro-military Japanese faction. And this is seen as one of the sort of lead-up events to the Second World War. Anyway, Katsuma has to go home to sort of take on some family responsibilities. In the meanwhile, he's sort of fallen in love with Gene Dan. And his older brother, who's the new Baron, actually has to come back and interview Jean and her family and her professors to determine if she's a suitable bride for his younger brother. He decides it's okay. It makes front page news up and down the East Coast. I guess it was an unusual thing, right? Japanese will marry Penn co-ed. Jean Clark had also gone to Penn, also is working with the same advisor. After they get married, they're both going to return to Japan and work at this new place called the Misaki Marine Station, mostly doing initially the kind of work that Katsuma Dan was doing. Now, the Misaki Marine Station was actually older than the Marine Biological Lab. It's based off the Naples Zoological Station. It's a beautiful, beautiful place just south of Tokyo. It's officially part of the Tokyo Imperial University system. It's right down here on this nice little peninsula. They're there in the sort of immediate pre-war years. The most famous story about this marine station is this sign. So when the Allied forces invade, they take over Misaki, and Katsuma Dan rides down on his bicycle and hastily writes this note and sort of pins it up on the door, right? That basically says, this is a marine biological station, don't destroy it, okay? Now, this may strike you as kind of a weird thing, and it always struck me as a weird note to leave on a, like, why would they automatically destroy a marine station kind of thing? Because it wasn't a marine station at that point. It was a Navy base for the Japanese Navy, and not just like a Navy base. It had these little tiny midget submarines called Sea Dragons. They're suicide submarines. So the people get strapped into them. It's basically like a giant motorized set of torpedoes. And their job was to, any ship that tried to enter Tokyo Bay, they were going to ram it and sink it, right? So it's not an insignificant military installation. So when the Navy, the US Navy comes in, it's not surprising that they might want to shut the entire place down. Basically, Katsuma Dan is able to keep it open and very quickly transform it back into a biological research station. They meanwhile had 5 kids living in Japan. They actually had to relocate up into the mountains where it was safe during the interwar period. And during this period, Jean Clark's research also sort of goes on hold. So what are they actually doing? From 1937 through the war, they're working on a set of papers on cell cleavage. Basically, they're just trying to describe what happens during that initial cleavage period. And they're using a variety of organisms to do it. So they start off using this very nice sort of blue tuxedo sea urchin. And the problem is, when you look at the egg under the microscope, it's clear. You can see it, but you can't see a lot of structure. So what they do is they get these kaolin particles. It's a very fine clay, and they coat the egg in it. And it makes little bumps. And so now what they've done is they've created sort of a marking system, so they can watch the membranes move by watching the motion of these particles. They then sort of branch out into sea dollars with that. The only one that they don't use that's from Japan is this little snail. They had done this together when they were at Woods Hole. And the reason they like this snail is that it cleaved very slowly, okay? And I'll show you why that's important in just a second. So this is the kind of thing they're doing. This is a series of drawings that's done with a camera lucida. So they're looking through the microscope, there's a mirror, and they're drawing exactly what they see. So they have to draw as fast as the cell is cleaving, right? And trying to be as precise as possible about the placement of those things. So a slow cleaving organism is good, right? Then you're more likely to be more accurate. And they really want to be super quantitative about this. They want to actually chart the distance that these particles are moving so that they get an idea about the spreading of that membrane during cell cleavage. Eventually, though, they're going to realize they have to use sort of different marker systems. They'll start to perforate the cell and follow the perforations around. And then by the 40s, they're going to switch to this beautiful little Hydra medusa. And this is in part because you can actually visualize the internal structure for the first time and see what's going on inside the cell. Also, the kaolin particles don't stick to it, so you have to sort of go that direction. So, what is the hypothesis? The idea is that spindles are important for pulling apart the cell. Today, that's like something you learn in 8th grade in the United States at least. They're the people that establish this fact. And so this is an actual physical model that they developed. This is a black ribbon, 2 little spools in the middle, and then bamboo sort of spikes. And what they would literally do is pull the spools apart and then look for slack in the ribbon to kind of measure what the dynamics were like. They started off with organisms that had central spindles, and then they moved to pick organisms that have acentric spindles to try to get different patterns of tension on that cell surface. And that's one of the reasons why they go with this Hydromedusa. It has extreme asymmetry in the spindles, and so this allows them, they think, to try to confirm this spindle structure. They think they can see the spindles in this diagram. Even if it were a clearer picture, it was like super hard to see those spindles. So why this new emphasis on cellular structures? Because the Cowlin technique doesn't work. There's another sort of breakthrough that's sort of lurking here, which is that Katsuma Dan takes in this young, skinny kid named Shinya Inoue. And Shinya Inoue is going to go on to become one of the world's sort of innovators in microscopy. And Shinya builds this microscope, which is called the Shinya Scope. They're all called Shinya Scopes. This one is based on a machine gun base that they found on the Navy station. And he takes a polarizing microscope here at the top, a T-can with a mercury vapor, and he's going to actually use the polarization patterns to find these patterns of birefringence in the spindles. And so you can see there's one spindle pattern here, there's another one down here, and that allows him for the first time to directly visualize those spindle structures. Okay? So this is where Katsuma Dan's research is going to go. He's going to start to use this polarizing technique to actually, instead of using marker systems, to now be able to use direct visualization of those cellular structures. Jean Dan kind of had her hands full with 5 kids, and then during the war and immediately after, she organizes their entire community relief effort. And she finds this deeply, deeply satisfying. It's a way of integrating herself into a community where she's not readily accepted, especially as an American in wartime Japan. Her husband gives her an ultimatum. He says, you must return to biological research. And he literally says, I will not consider you human again until you return to biology. So in order to do that, she returns to the Marine Biological Lab at Woods Hole and is talking with people there saying, look, I'm going to start my research program again. And they show her the newest, latest, greatest thing in microscopes, which is this kind of, it doesn't look like the newest, latest, greatest thing. It's a phase contrast microscope. And it's not just any one. It's the very first one built by Bausch and Lomb. Like it's the sort of sale version of this. They bring it down to the Marine Lab to show it off to make biologists buy it, right? And she sort of convinces Bausch and Lomb to sell it to her to take back to Japan, right? And it's actually a gift for her husband. He doesn't want it though. Instead, what he really prefers for her to use it to actually do research on her own. So what she decides to do is to solve or to examine this problem of fertilization. Specifically, what she wants to do is just answer this very basic question. When the sperm enters the egg, does the tail of the sperm also enter the egg? And you would think they would have solved that one, but no, it was not equivocally sorted out. But using this very nice phase contrast microscope, you can actually watch those fibers go into the cell, and the answer is yes, it all goes in. Okay? But even more, what she found is that during that period of agglutination, so remember Lily says when the sperm and the egg come together, they kind of react and form this globule. If you watch that process happen, you can see these sort of jelly coats around it, but you also see these really thin fibers popping out. And she notices that at the tip of the sperm, there seems to be a reaction and this fiber shoots out right at the point of contact. And so that's called the acrosome reaction. This is the acrosome. This is the acrosome filament. That filament is the thing that goes into the egg, and the egg then pulls the sperm in based on that acrosome. Okay? So because this microscope allows you to see those kinds of structures, she's suddenly able to understand what's going on during this agglutination period and what's actually causing that sort of attraction between sperm and egg at the time. This is a huge sort of advance in the way people understand fertilization. It's also a huge rejection of Lilly's research program because they realized that they shouldn't be worrying so much about the agglutination and really be focusing on these other subcellular structures. She does this primarily in these nice starfish that you get in the bay, which sort of readily available, very easy systems to use, very easy to sort of visualize under the microscope. But the very next thing that she does is then start to expand the list. So this is one of her review papers in 1967, and this paper primarily is just this giant list of all these other organisms that they've tried to establish this in. And so it's an inductive program, right? I found it in this starfish. What's the next thing I'm going to do? Look in sea urchins, and then look in sand dollars, and look at everything. The list is almost all marine organisms, because marine organisms are what they have available and what they're used to dealing with. It starts to expand out to fish, and you notice there's a little bit of mammals down here, some rabbits, rats, a gibbon even. This is much harder to do because these are internal fertilizers, right? And so that whole process is just a much more difficult process. And these are done primarily by Europeans and Americans, whereas almost all of these are coordinated through Japan. The acrosome reaction really makes a huge splash. It becomes its own object of research. This is a picture of a group meeting in Sweden working on it. This is Gene Clark in the middle. And these are the colons out here. This is Asvalius, who will go on to work on this as well. So this is really sort of re-energizes Gene Clark's career. It allows her to get a professorship, which was a very rare thing for an American woman. She's teaching at a women's college named Ochinomizu, which is north of the Marine Station, and becomes sort of enshrined and much loved. That picture of the microscope, that's like in a little memorial to her. There's been 3 biographies written her in Japanese. I mean, she's sort of, in some ways, has a better reputation than her husband because of that kind of research. Now let's focus a little bit on these two, the Colwins. Arthur Colwin, born in Australia, comes to the Second World War, is in Canada. He's actually named Arthur Cohen. And thanks to finding his military records, I can tell you he changes his name to Colwin during the Second World War, right before he immigrates to the United States to start working at Queens College in New York City. They know the Dans. I mean, it's a small world. They're keenly interested in those questions of fertilization, but they are approaching it in a very different way. The technology of choice for them is electron microscopy, which is sort of a much bigger, newer technology. Much more difficult to use, though, than those phase contrast microscopes. And the organism that you pick is a marine organism. It's this ugly acorn worm. And they're studying the exact same process, the exact process of fertilization where that sperm meets the egg. These are drawings of these incredibly beautiful micrographs. I want you just to look at these pictures for a minute. This is super clear. So here's the egg, here's the head of the sperm coming in and touching the egg. How do you make that picture? It's an electron microscope. So you have to get that egg and that fertilization moment. You then get the whole thing fixed. You have to slice it exactly. You then coat it in metal, and then you look at it. Okay? How many times do you think they had to cut an egg to get exactly that plane of fertilization, and then get lucky enough to actually coat it correctly and see it?
Thousands.
Right? Thousands. This is really extraordinarily hard to do. Okay? That difficulty explains why they're using that organism. They're able to make this organism polyspermic. In other words, it can be sort of fertilized by many different sperm, which reduces how much cutting they have to do to actually get one of those nice pictures. Their entire research program had a very familiar rhythm to it. So these worms are available at Woods Hole. They are gravid, they're ready to fertilize in the spring, in April and May, and then again in August. You can collect 50,000 eggs and embryos within those 3 weeks. You can use some temperature-induced ovulation as well. the organism will get standardized as they go through their research. Now, they produce a whole series of papers. All of them have this sort of pace. They're at Queens College, which is a teaching-heavy college. They're teaching 3 to 4 classes per semester. They don't do any research while they're teaching those classes. They get out in late April. They go to Woods Hole immediately as those acorn worms are fertile. They collect the fertilized eggs, and then they spend the rest of the summer trying to get a good picture, right, with those fertilized eggs. There's no electron microscope at Queens, right, so they're using just the equipment down there. They have to finish it up by August, so they get their pictures and their results. They bring it home in August. Every paper kind of gets done in September, right? So they write up their results, send it in, gets reviewed, and published sometime, within the next couple months, and then the whole cycle starts over again, right? So in terms of organism choice here, they picked an organism that works with their life cycle, right? Their life cycle as academics is, I need something that's ready to go in May, finish it up by August, write it up, publish it, and do the whole thing over again, right? That's as important as material availability, phenomenal access, all those other things. It works with them as much as they're working. Okay? So if we sort of back out to that big picture and go back to that organismal choice matter, organism choice definitely matters. It's definitely not a matter of convenience. Instead, we have all sorts of other things that enter into the question. So I would say organismal choice then is a complex issue. We would expect that as we study more and more people, we're going to find different sort of weightings of those values, different ways that those are instantiated. It's highly context dependent. Depends on the problem, where you're at, what kind of institutional support, what kind of community you have. And in some ways it's just contingent, right? They got lucky that they were able to find, the Colwins did, that they were able to find a polyspermic marine organism that fit with their life, right? And so in some ways, August Crow is great. It's a nice little principle, but the reality of organism choice is certainly going to be much more complex and a lot more interesting for us to study. Thanks a lot. Oh, here's
questions.
Rolling.
Yeah.
So, where do you stand on the either of two extremes of what might be called the porcupine studies, where we studied this animal and then this animal and then this animal and then now we'll study the porcupine, which is just doing more of the same. On the other extreme is the underrepresentation of the diversity. I mean, is this going towards some sort of a the case studies lead to a directive?
I'm reluctant to be directive. So here's the sort of the next question that I'm going to ask, which kind of gets at this. If you start to look at, so this sort of takes us right up to about 1968-69 in cell biology. They're still doing comparative research, right? So all of them are like, I found it in this system, then I'm going to do this system and this system. So they're building inductions based on diversity of cases. Starting in the 70s, many of their colleagues who are studying fertilization take it molecular. And one of the interesting things that happens is that the comparative aspect just drops out, right? And so the problem I'm trying to solve next is why? Like why are cell biologists comfortable with that? Why are they comfortable not comparing it to any other species? So that presumption of generality seems to become entrenched starting in the 70s, and I'd say it's still very strongly entrenched in cell biology. So one of the things we're trying to do is actually now go through the cell biology literature and actually document that loss of diversity in cell biology, and then to try to figure out why. Why did that?
By entrenchment, I'm sorry to pick up that, but by entrenchment you mean that they're focusing on the binding properties of a certain class of molecules, and once they find that, they're done and they don't need to see
that
where it works?
They're focusing on a binding class of molecules that they think works for all molecules of that kind based on studies from Drosophila, right? So in other words, they're not going to go, oh, we found, this kind of adhesin in Drosophila, let's go see if it's the same in mice. You don't need to do that, right? You find one, you found them all. Right? You study DNA in one organism, it's the same. Central dogma is the same in every organism, right? So where does that faith come from? They didn't have it. right? They were keenly interested in doing this comparative analysis. So, Uta?
Have you found, looking into the development of research on model organisms, have you found an impact of big data research on this research? I talked to a couple of people of Caltech and others, and they said it is sometimes increasingly problematic to receive funding from NIH because they said we don't need even mice research. We have now big data, we do that without model organisms. Have you seen a development or decline of research on model organisms?
This is one of the things we're tracking. So part of our sort of explanation for success was that they developed the big data databases, and those databases have the data, the annotations, and all the publications curated together. So it's kind of like one-stop shopping that's supposed to make that research easier. So if you go to like TARE, the Arabidopsis or Flybase, like all the information's there. The big question that is not necessarily that big data is going to make all that research obsolete, but like CRISPR-Cas9 is going to sort of start cutting into the need to do model organisms. So with CRISPR-Cas9, you can do gene editing in any organism you want, and so we're no longer going to have to just sort of stick with Drosophila where we know its genome exceedingly well. But it's still early days yet to actually see if that pattern. But we're patient, you know. We'll count. It's all right. We'll figure that out.
What about the idea that, I mean, I know that there's a push here I mean, it's very incipient, but there are a few people who are trying to convince our Institute of Health, National Institute of Health, to do away with animal stages.
Normal stages?
Yeah.
Why?
Because they're so badly predictive of what you're going to find in humans.
Wow.
And sorry?
No, this is part of the propaganda of the big data people. They say it's not predictive, and so we can just get rid of them. We just go on NIH,
Yeah,
for example, go on the big data research.
I think on average for a drug that you want to develop, that takes you back 7 or 8 years, and between $1 billion and $1.5 billion.
Yeah.
And the predictive value is extremely low. It just seems like an impediment. And if other countries outside of the U.S. are able to convince their national institutes of health to go for it, then the NIH will need to, to react.
Yeah, one of the things that justifies those choices of model organisms is that sort of human organism analogy. And so one of the first databases that's now obsolete was called Homophila, like Homo sapien Drosophila, and it was all the exact analogs between Drosophila and humans. And that's sort of, that's all the database showed you. It was like, here's the parallels between the two. And that's kind of defunct, has been rolled into other ones. Yeah, so if it undercuts that argument, that's the principal argument for the modelness of these model systems. So it'd be interesting to see if they do that. I mean, there's all those ethical issues about drug testing the other way.
Erez, what organisms do you work with?
Actually, I'm doing parallel. Actually, now it's an octopus.
And why did you choose the octopus?
Because the phenomenon we work with is extremely rich in octopus. Something very, very unique for octopus.
Yeah, so it's like the Croc Principle. Like there's certain things that certain organisms are just great for study.
But do you sleep well at night with the notion that whatever you find in the octopus will be
We know it's specific for the octopus.
No, but in terms of the ability to extrapolate to other organisms.
We know we cannot. Actually, this
You know you can't.
Yeah,
Right.
we actually want to understand why it's so unique in the octopus in order to actually try to do some engineering out of this, because it's something protein with very unique ability to do to change the RNA. And if you can use it, you can do something like the CRISPR on the RNA. But this is the idea.
Right.
Yeah, there's no sort of normative part to it. I mean, as a biologist, you have to sort of pick which direction you're going to go. Are you going to go with the pack, or are you going to, strike out on your own with something? And physiology has a tradition of going for the unique. They go for extreme adaptations because those are the the most striking cases. Right? So nature has done this job of creating a unique physiological system.
Mm-hmm.
Have you tried any other quantifications of success?
Of success?
Yeah.
We tried actually to get all of the funding data, and we almost had NSF give it to us, and then they talked to each other about it, and they decided that they wouldn't release that information. Because they had to search it themselves, right? They release information, but it's not in a, it's deliberately, I think, not in a usable format. Like you can't search by organism through their funding database. So they're required by law to release which grants get funded. But yeah, it's not the easiest system to work with. So we asked them to search it. They searched it. They said, wow, this is really interesting. And then they never gave it to us.
Let me put it differently. If you'd have your pick
Yeah.
onto what would be the metric to define success other than monetary?
Then money? Well, we think it's these repertoires. We think it's actually infrastructural development that actually seems to be making the most successful systems. Now that's the big systems, right? That doesn't mean that studying this octopus is not successful. This is almost like a problem-solving metric, and that would be a really great intellectual way to go. But how do you quantify like how many problems got solved with Drosophila this week versus and not every paper solves a problem, right? So yeah.
Do you find that in different strands of biology, so for molecular phylogenies, the ability to create these trees is an ability that was evolutionists were very happy to suddenly for this technology to fall into their lap. Do you think that a success in one field in biology can sort of be a good argument for people for a community in a different field of biology to be taken in by, so it does really well in evolutionary phynologies, really help us, make a lot of sense out of things that were totally, unknown to us. So now, for that reason, we'll tend to believe that for answers and for questions in basic molecular biology or physiology, using this approach is useful. And maybe that's a mistake. Maybe that's actually a mistake. That you're thinking it's successful in one field, and so it's going to be successful in mine. Maybe that's a mistake.
Well, because phylogenies are inherently comparative, right? So it has to do. And so when we do the journal Biodiversity, ecology is of course much more diverse. Evolution is much more diverse. But then it feels like neuroscience, where it's very narrow. Cell biology, I think, is going to be very narrow. Yeah. We did genetics because the whole thing comes out of genomics. So the whole idea was, there's another way of saying it has this genomic project, sort of narrowed genetic research. And it has a little bit, but not as much as people say it has.
I have a weird question.
All right.
I'm out of my homeland. Do you think that, or do you know that most of the researchers in these fields might be male?
Like, what's the sex breakdown?
Yeah. I mean, sensitivity to diversity is
Sure.
there might be a connection.
So I have a whole presentation on it. So this is the General Embryological Information Service. These are what the pages look like. So this is the developmental genetics page, and then they have listings of every single person in here.
Yeah.
And then the letters are their organisms. Because they're sexist, in the directory part, they put a Mrs. or Miss next to every woman's name.
Okay.
And so that meant I could have undergraduates sex every researcher in the database.
And if she's a professor, also Mrs.
Oh yeah. Oh yes. Yes. And so we've been able to sort of look at the gender breakdown.
Who's married also.
And who's a couple, because that's also a very common pattern that you come into science. That's right.
Yeah.
And that was deliberate too. We talk about how those women got into science and then maintained careers. At this time, that was the primary path. So when we do that, we did it, and also broke it down by nation. Different countries have different patterns. Most of the people in here are in the United States because of post-war investment. That's just where most biologists are. It's not nearly 50-50 in the United States, though. In France and Italy, it turns out to have a much higher proportion of women. It's almost almost exactly equal. But then the other thing you can do with this data set is it actually tells you what your job is, because it gives every person's position, what lab they're in, and things like that. So then when you do a division of labor analysis, like who's the professor, who's the associate, who's the technician, all the women are technicians, right? None of them are in the professoriate class. And that's also exactly what we would expect. Now, I thought France was like really like liberal and ahead of it because they actually have a higher number of women professors in certain labs. And I thought, oh, progressive French, how wonderful is that? And then I dug into like what it actually was happening in that lab, and it turns out that what was happening is that the lab was training equal numbers of men and women. The men could get jobs, and the women could not. And so the women were simply being retained in that laboratory. So it wasn't like a super liberal, progressive idea. It was just that they could not get placed.
How did they explain that?
What do you mean? Why
That the men can get jobs and the women.
I don't. They didn't
They
explain
didn't.
it.
They didn't.
I think
They
it's
didn't
just.
have
I think yeah. It's like,
No, they wrote everything. So.
well, no.
This is the fact, and live with it.
Yeah. So I thought that the one it was Etienne Wolf's lab, and I just thought he was like a very progressive person, collecting these brilliant women. Like Douarin came out of his group, and she did this amazing work on chimeras. And I thought, wow, he's just like gathering all these great women there. Like no. Like he's just stuck with all the women that can't be.
I have this follow-up question, and I'm asking you. I think it will be intuition of you. Do you think that this generalization assumption that the biologists have is related somehow to that gender issue? Because gender, I think when you deal up with gender, and I deal up with it a lot, you turn to pay attention more to the diversity between things and species and cultures and groups and people, and when you don't have to, so you don't have to.
I don't know the answer, but I could do the research and get you the answer to that question, because we have the people sexed, and then we can actually see who's doing multiple organisms versus who's doing single. My hunch about why the presumption of generality becomes so entrenched is that it's molecular biologists' fault. And remember, a lot of the early molecular biologists were physicists, and they deliberately simplified biology into its most sort of reductionistic form. They were studying bacteriophages, the simplest systems they could. And I think they didn't, first off, they didn't have an appreciation for sort of natural history and diversity. And I think they were very happy to generalize because they saw that as the successful way of thinking as a physicist, right? You simplify, you create a model, and then it explains everything until it doesn't, right? So that you deal with diversity after you.
And they are also mostly male.
Almost entirely.
Okay.
Yeah. But, you know, I don't think anybody's ever asked that question either. Like, I don't know of any of the physicists and like, that's an empirical question. But I think, yeah, they're going to be almost all the ones that are most famous are definitely men. So.
That would be fascinating.
Can you offer just a more like a more general claim for this research that has to do with you know, how we think about science more generally? Because obviously, you know, the general public thinks that the scientists sort of, you know, go to nature, look at what's there, you know, do their experiments, deduce their find their solutions to the questions that they ask. And here, what you're showing obviously is that that doesn't actually work that way. That the way we approach nature has a lot to do with, you know, us being human beings, with all our foibles, our personal histories, our biographies. You know, the fact that we're a social species, and so we create networks. And so I invite you to give us your take.
Well, I think that's absolutely correct. Yeah. So yeah. Yeah, so the whole project is based on that deep, and organism choice, problem choice is one of those. That's part of the contingency. It's in some ways very personal, but also very much a matter of which lab you happen to get into and what it happens to be like, and I think what your discipline happens to be like. So it's a profoundly contextual, social part of science.
Right. And then sort of the interesting work of the historian is to show how it's how what we know about, or it's difficult to do because you can't point out all the things that haven't been done. That's the virtual history part
Right.
we were talking about the other day. But it's an interesting question. How is our view of, basic fundamental mechanisms in basic biology to begin with, but then everything from that and onward, how is that skewed by the fact that we are human beings acting in particular networks through particular biographies in particular times and places?
I mean, yeah, so for a historian, historians tend to write about famous people, like people who get big prizes. And so the way we're conceiving of this project is that it's history from below. We want to write the history of every developmental biologist in the 50s and 60s. Like all the people, not just the people who won the Nobel Prizes. And asking a question like this, like how did you pick your organism, we think is a significant question, but it doesn't have to be answered by the guy who won the Nobel Prize. You can pick any of them, right? And you just have to get lucky and find someone that you have enough information to be able to tell a rich and complicated story about those things. It doesn't have to be based in the United States. It doesn't, it doesn't have to be a man. It can be anyone. And so slowly but surely, we're building those profiles, and I think it's a much richer, much more interesting sort of depiction of science. So I didn't think of it in advance. I could have just used all Israeli examples, because they're also listed out by country, and we've got all of those people listed too. So eventually, I'll get enough collaborators. Well, everybody will have a country and sort it all out.
Is there a supply side story to tell as well as the demand side about this? Like when DuPont starts marketing Uncle Mouse as then they're clearly marketing, but is there a prehistory of that kind of thing? Are there companies that are selling these who are marketing in clever ways and that influence?
The Marine Biological Lab is a biological supply company. Yes, so they you write to them and say I need sand dollar eggs or whatever, and then you'll get a little box on the train the next day.
But did they ever try to influence the choice of organism? Did they have more profitable organisms to sell, or?
That would be a good question. Yeah, there definitely were networks of supply, but they usually, if you're doing embryology on amphibians, it was kind of like who you knew, like who could get you the amphibians and things like that. And they often had to be very local because these things would die if they were too far away. So, for instance, Drosophila couldn't be shipped by train because the train would almost always go through Texas, and there was no air conditioning, and they would all die by the time they got to the East Coast. And so the Drosophila network is very, very constrained until you get multiple centers of supply. Then, in fact, we can trace Drosophila going from the U.S. to Europe by Herman Muller taking an ocean liner with a bunch of bananas and bottles of flies, and in his stateroom, like mashing, like you can literally follow these organisms
feeding the flies
around the world. That's how they get to Berlin, then they get to Moscow. They come to this region of the world when a woman actually comes in the 1950s. Her husband is a naval officer, a physician in Beirut, and she brings Muller's flies to Beirut and then starts trapping flies from Israel and Lebanon. And so that's literally the route of transmission.
I have another weird comment question. Probably completely off, but you got me thinking about the limitations of the use in animal models in psychiatry, which is one of the biggest challenges, perhaps, to continue with the organism. So, if you have any...
Yeah, I don't know much about that.
Okay.
I have colleagues that are definitely studying that. And in fact, we just had a paper last week at Pitt about people who were trying to have human-animal systems that mimic each other. And so they had tests for cognition that are based on touch screens for humans. And so it was about the creation of touch screens for mice. where they touch their nose to it. So it was like a fascinating study of trying to bring these systems in exact parallel with each
So
other.
they start with the normal functioning.
That's right.
So what is like ADHD in a mouse?
Yeah. Well, people who couldn't make the pattern comment, the pattern connections, they thought were like representative of schizophrenia. I mean, it's very
Really?
tenuous, I thought. Yeah.
Wow.
There's also the issue of maybe it's way off, but like morality of use in animals
Yes.
for medicine research. So maybe your thoughts on this.
Yeah, so this is
highly
part of
problematic and important question.
Yeah, this is part of an Australian Research Council grant, and part of the Australian group is doing animal ethics questions explicitly within the Australian context, and also trying to understand this question of why Australian marsupials aren't widely used research organisms, even within Australia. But yeah, the animal ethics questions are very interesting, especially we're very interested in different ethical norms in different countries, and why those aren't sort of more uniform, and then what that process of communication and agreement is. And we're sort of very interested in these cases of what I call biological rendition, where you're not allowed to do something to an organism, but you have a friend in another country where they have a different ethical standard, and so you just become a collaborator with them, and like, you can do that to those rats, right? And they do it, and so you're sort of going to the least common denominator, which is not what you're supposed to do, right? But that's what happens, and they can say, well, it's still within the ethical constraints of that country, and that's where the poor rats were, experimenting.
Can you give us some striking examples of differences in ethics between countries?
Like highlights.
Graduate students got the list of that. That's not in my head at this moment.
Yeah. All right. Any other questions from the audience? Okay. So thanks a lot, Mike.
Thanks.